Welcome to all of you to biol4u.com. Today I am going to discuss a biosynthetic pathway of photosynthesis. We already discussed photosynthesis is a biochemical process, physico-biochemical process in which there are two phases, that is the light phase and dark phase. In the light phase we already discussed during the light phase the assimilatory power, that is the ATP NADPH is prepared, along with the oxygen is also formed. This oxygen is released as the byproduct. Okay, and this ATP and NADPH is used for the biosynthetic pathway, and this biosynthetic pathway is also known as the dark reaction. Let us analyze how the biosynthetic pathway proceed, what are the different aspects of this biosynthetic pathway. So in this video we are going to answer some of the questions, that is the where are the ATP and NADPH is used. Okay, this is prepared in the light reaction. Okay, so where it exactly used in the biosynthetic pathway. Okay, so which molecule is the first stable product in the carbon dioxide fixation? That is, when carbon dioxide absorbed by the plant, which chemical is the first product formed after the carbon dioxide absorbed and fixed? Okay, then the next question is, what is the primary acceptor of the carbon dioxide? That is, carbon dioxide present in the air, it enter into the leaf through the stomata, and who will accept, which molecule accept this carbon dioxide at first? Okay, then the next question is, who deduced the pathway of the carbon in carbon dioxide during its fixation? That is, when carbon dioxide enter into the leaf, it absorbed by the first acceptor of the carbon dioxide, and then the carbon in the carbon dioxide moving through the different intermediate product, and then finally we will get the carbohydrate. So the pathway of this carbon in carbon dioxide is deduced by a scientist. Who is that scientist? We will discuss later. Okay, so next question is whether all the plant following the same pathway for carbon dioxide fixation or not. So all these questions we will answer in this video. So let us begin biosynthetic pathway of the photosynthesis. Okay, so we learned that the light reaction produced ATP. So light reaction produced ATP. At the same time it also produced NADPH. These are the product of the light reaction. So along with it also produced oxygen. Okay, so I told oxygen is released out through the stomata. Okay, so and carbon dioxide taken inside, oxygen is released out, but ATP and NADPH not released. It will retain, and that later used for the biosynthetic pathway. Okay, so that is a relation between the light reaction and the biosynthetic pathway. So whatever the product produced in the light reaction, that utilized for the biosynthetic phase. So this biosynthetic phase is also known as the dark reaction. So it is also known as the dark reaction. But this term dark reaction is actually a misnomer. Okay, so it is a misnomer. Why it is a misnomer? Misnomer means the meaning, the word is one, but the meaning is different. So okay, let us see why the dark reaction, the term dark reaction applied for the biosynthetic pathway is a misnomer. Okay, misnomer means the word one, but the meaning is different, because when the plant produced the food, that is a carbohydrate, and this carbohydrate will utilize. Then if the plant not getting light, the biosynthetic pathway, that is the carbon dioxide, then again if you give the light to the plant, so the synthesis will start. That means light reaction is very essential for the synthesis of the carbohydrate. So during the light reaction it produced ATP and NADPH that utilized for the biosynthetic pathway that is taken place independent of the light, not in the absence of light, independent of the light, and that carbon dioxide utilized by the plant. When the carbohydrate utilized by the plant, and when the carbohydrate is completely utilized, the process of the photosynthesis will stop. Then again, if you applied the light on the plant, the light reaction will start, and again it is producing ATP, NADPH, and the dark reaction or the biosynthetic pathway will operate. So that means the biosynthetic pathway not only operating in the dark, but also it operating in other condition also. But during the dark time, when the biosynthetic pathway is operating, if you applied the light, even in the night or even in the dark, the plant will proceed light reaction. So it is not a dark reaction. The reaction taken place independent of the light only. So that is the term dark reaction is a misnomer for the biosynthetic pathway. Okay. So let us see how the ATP and the NADPH, that is ATP and NADPH formed in the light reaction are used in biosynthetic phase. How it is used. So we saw earlier that carbon dioxide combined with water molecule and produced a carbohydrate, that is the CH2O. So it is the representation of the carbohydrate, okay, or it is known as the sugar. Carbohydrate, or it is known as the sugar. So like that, sugar is formed. It was of interest to scientists to find out how this reaction proceed, how the reaction is proceed. It was a dilemma for the scientist for a long time, or rather, what was the first product formed when carbon dioxide combines with water. So which chemical was the first product? We written a general formula, that is CH2O. Exactly what is the first product of the photosynthesis or after the carbon dioxide fixation? You know, after the World War II, that is the World War II. After the World War II, scientists was very busy to find out the way how the radioactive isotopes or radioactive element can utilize for the human welfare. How the radioactive element can be used for the human welfare. During this period, Melvin Calvin, a scientist, Melvin Calvin very effectively, very practically used radioactive isotope, that is the carbon-14, to find out the pathway of the carbon in carbon dioxide during the fixation of the carbon dioxide and to produce a carbohydrate or sugar, So he was the first scientist used the isotope of the carbon-14 to find out the pathway of the carbon in carbon dioxide during the fixation of the carbon dioxide to produce sugar. Okay, so that was the use of the radioactive isotope after World War II for the human welfare. And by using this doing this experiment with the radioactive isotope C14, Melvin Calvin find out the detailed pathway of the carbon in carbon dioxide, and hence the cycle that is discovered by the Melvin Calvin, later known as the Calvin cycle. Okay, so this Calvin cycle explaining how the carbon dioxide fixed and how the carbon dioxide converted into sugar. So that cycle is known as the Calvin cycle. Okay, discovered by Melvin Calvin. And later Melvin Calvin got a Nobel Prize also for this discovery. Okay, so according to Melvin Calvin, the first stable product formed after carbon dioxide fixed by the plant is the 3-phosphoglycerate. Glyceric acid, 3-phosphoglycerate or 3-phosphoglyceric acid. 3-phosphoglyceric acid. Glyceric acid. You know, this phosphoglyceric acid is also organic acid, also known as the glycerate, means 3-phosphoglycerate or 3-PGA. That means 3-phosphoglycerate or 3-phosphoglyceric acid. Then how many carbon atoms does it have? How many carbon atoms present for this phosphoglyceric acid? When we analyze it, it is shown that the 3-phosphoglyceric acid is actually a 3-carbon compound. It containing 3 carbon compound. So scientists also tried to know whether all the plant having PGA as the first product of the carbon dioxide fixation. So Melvin Calvin obtained the result by hard work, that is the first stable product of the Calvin cycle is the 3-PGA. It is a 3-carbon compound. So the next question in front of the scientist is that whether all the plant having a 3-carbon compound as the first stable product after carbon dioxide fixation. No. Then after analyzing number of the plants, it came to know that some of the plant having 3-carbon compound as the first stable product and some other plant having 4-carbon compound as the first stable product. So you know this 3-carbon compound is the 3-PGA. Okay, this 3-carbon compound is the 3-PGA. PGA means phosphoglyceric acid, phosphoglycerate, and this 4-carbon compound was again an organic acid that is known as the oxaloacetic acid. Oxaloacetic acid. Oxaloacetic acid, or it in short it is known as the OAA, oxaloacetic acid. So some plant 3-PGA, 3-carbon compound is the first stable product. In some other plant it is the 4-carbon compound. So the plants are different type. Those plant that are having the first stable product 3-PGA is known as the C3 plant. So it is known as the C3 plant, or it following C3 pathway plant, or it is known as the C3 plant. At the same time, if the first stable product is this carbon form, means 4-carbon compound, then it is known as the C4 plant, or it is known as the C4 pathway. So plant preparing the food, that is the carbohydrate, by two pathway, that is the C3 pathway and C4 pathway. Some of the plant majority of the plant doing C3 pathway, and some of the plant doing C4 pathway. We will study the C4 pathway in detail later. Okay. So then next, so we know it concluded that it become very concrete that the first stable product is the 3-carbon compound. Majority of the plant, C4 plant, that we will consider later. Okay. So 3-carbon compound, 3-phosphoglyceric acid or glycerate is the first stable product. It is confirmed. So the next question is that who is the first acceptor of the carbon dioxide? Who accepted the carbon dioxide first? So let us see which chemical, which molecule first accept the carbon dioxide. You know, carbon dioxide fixation taken place and the first stable product formed, that is the 3-PGA. But which molecule first accept the carbon dioxide? It was a very painstaking, after very painstaking experiment, scientists discovered the first chemical that absorbed fixed carbon dioxide. Okay. So the primary acceptor of the carbon dioxide. Let us see the primary acceptor of the carbon dioxide. Primary acceptor means the first acceptor of the carbon dioxide. Primary acceptor of carbon dioxide. So it was a very difficult to find out which molecule first accept the carbon dioxide. So you know the first stable product is a 3-carbon compound. So most of the scientists believed that the first chemical that absorb fix carbon dioxide must be a 2-carbon compound. So 2-carbon compound plus 1 carbon from the carbon dioxide, and it produced the 3-carbon compound. It is very simple. But this finding, this arbitrary finding, imaginary reaction, it is not a correct, because after many decades of the experiment, many years of experiment, scientists came to the conclusion that the first chemical that absorb fix carbon dioxide was not a 2-carbon compound, but it was a 5-carbon compound. So this 5-carbon compound fixed carbon dioxide and produced the first stable product, that is the 3-PGA. 3-PGO. Here. So the simple concept that 2 carbon plus a carbon gives a 3 carbon, it is not correct. It is absolutely wrong. So, but the scientist previously believed that it may be the correct. But the very painstaking experiment, many years experiment, scientists came to conclude that the first carbon dioxide fixer is not a 2 carbon compound, it is a 5 carbon compound that combined with the carbon dioxide and produced 3 carbon compound, that is the 3-PGA. Okay, so the next we are going to explain the exact Calvin cycle. So up to that we know that the first stable product is a 3-PGA, 3 carbon compound, some of the plant 4 carbon compound oxaloacetic acid. First, the chemical that fixes the carbon dioxide is not a 2 carbon compound, it is a 5 carbon compound, and the product of the light reaction, that is the ATP and NADPH, used for running the biosynthetic pathway. Biosynthetic pathway is also known as a dark reaction, but it is not, it is a misnomer, because during the dark time if applied light, that also following the light reaction and proceed the biosynthetic pathway. So it is only, we can conclude biosynthetic pathway is independent of the light, and light reaction is depending on the light, or light dependent reaction. So now exactly what is the Calvin cycle, how the Calvin cycle is operating. So let us see the Calvin cycle, how the Calvin cycle is operating. So the Calvin cycle. It is known as Calvin cycle because it is discovered by Melvin Calvin. Calvin cycle. Okay, Calvin and his co-workers finally worked out the whole pathway of the pathway and showed that the pathway operated in cyclic manner. Okay, so the carbon moving in a cyclic manner. It is starting from a point, this point, and then finally it reaching to that point. That means the pathway of the carbon in the carbon dioxide during the dark reaction or the biosynthetic pathway is following in a cyclic manner. Okay, then the first carbon dioxide acceptor, that is the RUBP, rubulose biphosphate. This is the first carbon dioxide acceptor. It is a 5 carbon compound, not a 2 carbon, 5 carbon compound. Okay, so this cycle starting from the RUBP, at this point RUBP, and then the cycle operator, and finally the RUBP regenerate. So hence this process is in a cyclic manner. Okay, that is taken place in the cyclic manner. So let us now see how the Calvin pathway operate, how the Calvin pathway is operate, and where the sugar is synthesized, how the Calvin cycle operate, and at which point the sugar is synthesized. So let us at the outset understand that very clearly that Calvin pathway occur in all photosynthetic plant. First point, very important point, whether the plant is C3 plant or whether it is a C4 plant, Calvin cycle is operating in both. Means in all photosynthetic plant, Calvin cycle is operating. Compulsory Calvin cycle is operating. But the anatomy of the C3 plant is a little different, and the anatomy of the C4 plant is a little different, because to operate a C4 pathway, it requires some additional characters, and besides the characters present in the C3 plant. But in both the C3 plant and the C4 plant, the Calvin cycle will operate. Okay, so for the easy understanding the Calvin cycle, we can divide the Calvin cycle into 3 phases. That is the phase number 1, it is known as the carboxylation. Carboxylation means addition of the carbon dioxide, that is the carboxylation. A molecule become carboxylated, that is the first reaction. The second reaction is the reduction reaction. After the carboxylation, it produces the first stable product, that is the 3-PGA glycerate, and that glycerate undergoing reduction, and after the reduction the last stage is the regeneration. regeneration. What regenerate? RUBP regeneration. So reduction, regeneration. So these are the three stages in the photosynthetic pathway. So let us explain these three stages separately. So first, carboxylation. Carboxylation. So what happens in the carboxylation? Actually, carboxylation is the fixation of the carbon dioxide fixation into a stable intermediate. The stable intermediate is the 3-PGA, 3-phosphoglyceric acid or glycerate. Okay, carboxylation is the most crucial step. This step is the most crucial in the Calvin cycle. Okay, where the carbon dioxide is utilized for the carboxylation of the 3-PGA. So 3-PGA undergoing carboxylation of the RUBP, not the 3-PGA, RUBP. This RUBP, ribulose biphosphate, undergoing carboxylation and produce the 3-PGA, phosphoglyceric acid. 3-PGA will produce. Okay, so this reaction is actually enzymatic reaction. It is requiring an enzyme. The name of the enzyme is the RUBP. RUBP carboxylase. So which result in the formation of the two molecule 3-PGA. It produces 2 molecule 3-PGA. Okay, it actually producing 2 molecule 3-PGA. Okay, since this enzyme also has an oxygenase capacity. See, this enzyme also acting as an oxygenase. That time we can say that it is a RUBP oxygenase. Okay, that means this enzyme working both as a carboxylase and oxygenase. It is depending on the concentration of the carbon dioxide and oxygen. It turn to carboxylase or oxygenase. Okay, hence this enzyme is better known as the RUBP, ribulose biphosphate carboxylase oxygenase. Because both the property having this enzyme oxygenase. So in short, it is writing RUBIS, CO. RUBP carboxylase oxygenase. C means the carboxylase, O means oxygenase. Okay, so this reaction taken place and it is producing the 3-PGA. So let us show this process in a cyclic manner or in the form of an arrow diagram. Okay, so let us see this first reaction in the form of a arrow diagram. Okay, so first RUBP carboxylase, ribulose 1,5 biphosphate. So this absorbing the atmospheric carbon dioxide. So carbon dioxide with moisture. So this is from atmosphere. And this reaction is known as the carboxylation reaction. So this is the first reaction. Okay, it produced by some intermediate. It produced 3-phosphoglyceric acid. 3-phosphoglyceric acid produced. Okay, so this is the first reaction. It is known as a carboxylation reaction. So let us study the next process, that is the reduction. So how the reduction process is going on. So next, reduction. So definitely reduction means the 3-PGA undergoing reduction and producing the next product, that is the a triose phosphate. So by the reduction, that means 3-PGA undergoing reduction and produce a triose phosphate. Okay, and this triose phosphate is utilized for the synthesis of the food, that is the sucrose and starch. So this is the point where the food, that is a carbohydrate, is producing. So during this reaction, ATP is utilized. So ATP that is produced in the light reaction, that undergoing like ATP and also NADPH are used, and this ATP NADPH undergoing it is helping for the reduction, and ATP is converted into ADP, that is giving to the light reaction, and NADPH converting into NADP, and that also giving to the light reaction. So with the help of the ATP and NADPH, the 3-PGA reduced to triose phosphate, and this triose phosphate utilized for the synthesis of the food. So 3-PGA, it is producing triose phosphate. This triose phosphate used for the synthesis of the sugar, that is the sucrose and starch. Here the reaction taken place, ATP plus NADPH, so that oxidize and producing ADP plus NADP. Okay, this product transfer to the light reaction. Okay, so this is the third process, and this process is second process, that is known as the reduction process. So this is the reduction, second stage, that is the reduction. So this is the stage number 2. Okay, so then one more stage remaining, that is the regeneration. So what is actually regeneration? So during the regeneration, the 3-ribulose 1,5 biphosphate is regenerating. So what happens here? This triose phosphate, the triose phosphate prepared, that helping to regenerate ribulose biphosphate. So this is the regeneration stage. Regeneration. So that is the third phase of the Calvin cycle. So here 1 ATP is utilized. So 1 ATP is utilized, not 1 ATP is utilized, converting into ADP, and energy released, that energy utilized for the conversion of the triose phosphate to ribulose biphosphate. So we can divide the cycle into 3 stages. That is the 3 stages. That is the number 1, it is called carboxylation, and number 2, that is the reduction, and number 3, that is the regeneration. So the regeneration is with the help of the triose phosphate. So here produced. So this is the regeneration stage, this is the reduction stage, and this is the carboxylation stage. So by completing all these stages, the carbohydrate will prepared. Okay, so this is the Calvin cycle. So hence for every carbon dioxide molecule entering, each carbon dioxide molecule entering to the Calvin cycle, 3 molecule of ATP is utilized. Actually here 2 molecule ATP is used, here 1 molecule ATP, 3 molecule ATP is used, and 2 of NADPH are required. 2 NADPH is required. It is probably to meet this difference in number of ATP NADPH used in the dark reaction, that the cycle phosphorylation taken place. Okay, so to make 1 molecule of the carbon glucose, the glucose having 6 carbon, glucose containing 6 carbon. So to make 1 molecule of the glucose, the cycle must be operated in 6 times. So 6 carbon dioxide entering inside. When each cycle is operating, 1 carbon dioxide entering, so 6 times the cycle is operating, and it is producing the 6 carbon sugar, that is the glucose. So you know, for 1 cycle it is requiring 3 ATP. 1 cycle it is requiring 3 ATP and 2 NADPH. Then how many ATP and NADPH is required for synthesis of the 1 molecule glucose? So it is very easy, 3 into 6, that much ATP, and 3 into 2, that much 6, not 3, 6 into 3 for 1. So 3 into 6, and 2 ATP, 2 NADPH, 2 into 6, that much NADPH is requiring. So the total ATP is 18 ATP is requiring, and the total NADH, 12 NADH is requiring to synthesize 1 molecule of the carbon dioxide. Okay. So during this Calvin cycle, what is taken inside the plant or leaf, and what going outside? If we consider this one, 6 carbon dioxide enter inside and 1 glucose is out. 1 glucose producing, 1 glucose is forming. Okay. Similarly, 18 ATP is taken and 18 ADP is producing. ADP is producing, and similarly 12 NADPH is taking inside and 12 NADP is forming. So that is the Calvin cycle, the summary of the Calvin cycle. Then I will show the animation of this biosynthetic pathway, that is the Calvin cycle, how operating in the form of the animation. Okay.